The endomembrane system is a very extensive network of membranes that we find inside eukaryotic cells. And if we talk about what classifies something as being part of the endomembrane system, this is going to be membranes that are directly connected together, so in other words, we're saying that they're continuous with each other, or they can be connected by a transfer of vesicles. So if we talk about this a little bit more, the first thing I want to talk about is what we mean by vesicles. Vesicles is really a pretty generic term that's used to refer to, it's kind of just a bubble of membrane material. So this is going to be membrane on the outside, and as membrane, this is going to be a phospholipid bilayer. And then there's going to be space inside this. So all in here would be a compartment where we can have a number of different molecules located. So this is going to be on the inside, it's really a container for various molecules. And so when we talk about what a vesicle actually does, its main function that it's going to have is all about transport. And so this is going to transport things from one location in the cell to another. Or in other words, we think of these as cargo containers. So if the cell has something that it needs to transport from one location to another, typically it's going to place it inside one of these little cargo containers that we call a vesicle, and then it's going to send it on its way from one location to another. So let's take a closer look at the various parts that do make up the endomembrane system. So here we have a look at a eukaryotic cell. I've only put some of the components in here so far, but we have the plasma membrane around the outside, which would be found around all cell types, and then I've drawn in my nucleus right here. And recall that around the outside of the nucleus, just on the very edge, we're going to have the nuclear envelope. Now, as we start to talk about the endomembrane system, we will find that the nuclear envelope is going to be part of that, and that's because that nuclear envelope is going to really be continuous with what we call the ER. So this is all going to make up my ER. It's a network of membranes. So this is wrapping all throughout the cell, and we will see it kind of just a bunch of flattened sacs of membrane. Okay, so here we have just a whole bunch of it. And as I'm drawing it here, just to highlight what I'm trying to show, this is membrane on the outside. And so I'm just filling in, and then it has a compartment on the inside as well. And so again, let me just finish this drawing here so that we can all have an idea of what we're trying to show for this picture. There is a lot of interior space when we're talking about the ER, and there's a lot of surface area of membrane. This is considered to be where most of the membrane inside of a eukaryotic cell is actually located at. It's going to be inside the ER. Okay, so the blue, this is the endoplasmic reticulum, or we can call it the ER for short. And so when we're looking at this, it is actually directly continuous or connected together with this nuclear envelope, which is around the outside of the nucleus. Okay, so those are connected together. Now, one thing about the ER is that when you do have little things that are produced inside here, because it's producing all types of molecules, so we can look at those inside. Once they are produced, what's going to happen is a little vesicle, one of those cargo containers, is going to bud off. And so we're going to get this little container that pinched off, and it's got a bunch of molecules inside of it, and it's going to transport that vesicle. Okay, so again, just to emphasize what this is, it's going to transfer that vesicle to some other compartment or location where it needs to be. And typically where that is going to be is it's going to transfer it to the Golgi apparatus. So if we draw the Golgi right here, here would be components of the Golgi. The Golgi is not quite as big as the ER. It's quite a lot smaller. And so if we just draw it, it has a similar shape to it. It's a bunch of flattened sacs. Okay, so I'm drawing those here. And so this little vesicle is going to make its way to the Golgi. And when it does, it's going to fuse with the Golgi. And when it does, it dumps its contents inside. And so at that point, those contents will move through the Golgi. Again, we basically have little vesicles pinching off at different times. And then those vesicles come and they fuse with the next part and dump the contents inside. And when all is said and done, that vesicle is going to pinch back off of the Golgi, and it's going to have different molecules inside of it. So those would be molecules that have been processed a little bit more as they spent time in the Golgi. So if I label this here as our Golgi, notice that we are passing vesicles from the ER to the Golgi, and then the vesicles pass out. And then one common place that they will go at that point is a lot of times they will actually come to the plasma membrane. If that happens, then if we draw it down here, the vesicle is going to fuse with the plasma membrane. And I've drawn it a little larger just so we can see it a little bit better, but it's going to fuse with the plasma membrane. And when it does that, if we just look at what happens, the insides are going to be really dumped. So what was on the inside of the vesicle gets dumped or exported from the cell. So it's going to end up on the other side of the membrane. So this is one way that we can move different molecules from inside the cell to the other side of the cell's surface, so outside of the plasma membrane. So just to sum up what we're saying here with the endomembrane system, this is an example of it. The parts that would be included in this inside membrane system, they would be the nuclear envelope, because it is directly connected to the endoplasmic reticulum. So both of those would be included. We are going to include the Golgi apparatus as well, because the vesicles can move to that. And then those vesicles can also fuse with the plasma membrane. So even though the plasma membrane is on the outside of the cell, it's not technically endo or inside, we do include it in the endomembrane system. And then just a couple other generic components that could be included. The vesicles could be included. Other things, oftentimes we have smaller compartments, which we call vacuoles inside. Vacuoles are similar to vesicles, but these have more of a storage role. Okay, so I'm going to show them here, and I want to emphasize they are part of the endomembrane system because vesicles could fuse with those as well. And then one last one that we tend to have in animal cells is we can also have lysosomes, which can fuse with vesicles as well. So anytime we have something that can fuse with vesicles, or transfer vesicles back and forth, we're going to include it in that big endomembrane system or inside membrane system. This is something that you're only going to find inside eukaryotic cells, because in the prokaryotic cells, we don't have any inside membranes or internal membranes anyway.